meiosis, a review of what we talked about last time. What's the process of making gametes? Meiosis, right. Usually have the hint is the first thing is the title of that section. That's pretty easy. So meiosis, of course. 25. Blank refers to having two of every kind of chromosome. Diploid or diploid? I would just say diploid. And remember, we usually refer to that as being 2N. Okay. And so what refers to have only one set of chromosomes, half or N? In other words, haploid. Again, they didn't ask me. I should have been monoploid or uniploid, but they called it haploid for some strange reason. Number 27. Blank chromosomes are the matching set. They match up gene for gene. What do we call that word for when the chromosomes match up? Homologous. Homo meaning same. Homologous chromosomes. Not to be confused with homozygous. Be careful. Number 28. Blank is when there are four sister chromatids together. So when the pair is together like that and they saw four lines, what do they call that? The prefix means four. Tetrad. Number 29. Blank phase. When two pairs of chromatids come together during meiosis, sorry for all the spacing. When the pairs come together during what phase? Prophase 1. And during prophase 1, number 30 can occur when those tetrads cross and exchange gene for gene, providing genetic diversity. Crossing over. Remember, that happens during prophase 1. Number 31, blank is the phase when the pairs line up in the middle. It's still metaphase. The pairs line up in the middle, so it's meta which? Meta 1. Number 32 is how when one pair of chromosomes doesn't affect how the other pairs line up, leads to genetic diversity. It happens during meta 1, how the pairs line up, has no effect. Yep. Independent assortment. And Mendel saw that. He wasn't thinking about human chromosomes with linked genes. He just saw pieces. Number 33. Last one. Blank is when the pairs come apart. What? Anaphase 1. Ana apart. Anaphase 1. Pairs apart. Questions about this? Got it okay? Alright, so now you're keeping up with this blue sheet. You're getting a really good study guide for the vocab component of this particular test, which again is a week from tomorrow. Alright, so now, okay, deep breath. We're going to talk about our third twist on Mendel. Remember, we left Mendel a few days ago and his pea plants. And pea plants, remember, were just either tall or short, purple or white, wrinkled or smooth, or round, whatever you want to call it, yellow or green. So it was complete dominance. As long as you have one big allele, you had a dominant trait expressed. Well, we moved on beyond Mendel, and we had two little twists. First, we had incomplete dominance, where there was a what? Mixing, blending, right. Incomplete dominance, there wasn't a true dominance again, so we really didn't use big letters and little letters. We used different letters because the heterozygote had its own phenotype. So it was different. It was a little twist. So we had three options, three phenotypes. And then, of course, that changes up our, not necessarily our genotype ratios, but certainly our phenotype ratios. So the heterozygote had its own phenotype instead of adding to what the dominant one was. And then we had codominance. Our example being blood type. There are others, but that's the key one, where both traits are expressed at the same time. A's and B's are on the blood. Or if you saw maybe a poinsettia that had red and white. Or roan cows. I was talking about the roan cows where they have red and white hairs together at the same time. So now we're going to take one final huge giant leap away from Mendel and the pea plants. In fact, we're not even going to talk about horses or cows or any kind of flower. We're talking specifically humans today. Now, my little caveat to this, my little disclaimer before this lecture is, since it deals with humans specifically, some of the things that I may mention today may seem a little bit harsh or hard to hear because it is human and it has with us, and I get kind of close to home, but it is what it is. So it is life. It's life happens. So before we get to our class today, our third choice on theme, let's talk a little bit about human heredity specifically. Now, a karyotype is a picture of all the chromosomes matched up. All those homologous chromosomes that we mentioned in meiosis. What a scientist will do, or a doctor will do, will get a picture of all the chromosomes. So we'll define it first, and I'll show you a picture. This is a picture of all the chromosomes, all the homologous chromosomes matched up. I'm going to pause for a second. So this is an example of a karyotype. These are actual pictures of actual chromosomes. And generally speaking, a karyotype is usually done when it is thought that there's a possibility of something being wrong. Like I mentioned, this is not necessarily the easiest thing to hear, but generally speaking, around the age of a 35-year-old woman, most OBGYNs will ask, do you want to have an amniocentesis? What amniocentesis is, by developing with the baby, and they take in a long needle and they stick it into the belly. Doesn't hurt the baby. They let some of the amniotic fluid, right, that's floating around the baby. Of course, there's going to be some baby skin cells in there. And what they can do is take some of those cells, put all the chromosomes out. Those of you who are putting your heads down, today is not the day to be sleeping. Because what we're going to do with the cross is going to be a little more complicated. You don't want to miss that. And what they'll do is they'll cut them out and they'll match them up. Usually by size. They also, these are black and white, they usually have dyes too that helps to match up the genes. And what they do is they look for everything being there. This one is a normal karyotype. There's 2 1s, 2 2s, 2 3s, etc. The last set, notice that there's 22 sets of chromosomes that are actually homologous. These are called the autosomes, the homologous pairs. The last set, the 23rd set, we call the sex chromosomes because they determine the gender. And I know you know by now, if you have 2 X's, that is a female. If you have an X and a Y, that would be a male. So the kinds of things you can determine from this karyotype is that all the chromosomes are there, hopefully. You can see that all the autosomes are there, and you can see the gender of the baby. Can you tell if on the pair of 7s you've got, a dominant trait represented there? Can you tell that from looking at this picture? No, so you can't tell things like dominance, recessive, that kind of stuff. You can tell number of chromosomes and gender from a karyotype. Okay, so let's go. Okay, so you saw pictures. We'll see another one, an example. Sex chromosomes, like we said, it's a 23rd pair of chromosomes in humans. Okay, they determine gender. So XX is female, XY is male, and like I said before, that is why Henry VIII should have beheaded himself, because it was his fault. Males determine the gender. So a female egg is only going to have an X in it, as far as the sex chromosomes are concerned. A male sperm, if it's an X sperm, it will make a female, it's X and X. If it's a Y sperm, it will make a boy, XY. So the male determines the gender. All right, the autosomes are the other 22 homologous pairs. And that has all those other traits that we've been talking about so far, like hair color, whether or not home like this, or the tasters or non-tasters, and we'll test those with the next class. I've got the tasters versus non-tasters and colorblindness, we'll check for that next on Wednesday for sure. Okay, last but not least, as far as human heredity is concerned, is something called a pedigree. Now, you probably, do you have one on the board? No, you don't. You do have some room on the back. On the back, underneath, if you did your predominance, the blood typing thing, if you have notes on the back of that, you hopefully will have some notes at the bottom, because I'm going to draw a pedigree. I'm going to tell you the definition of the pedigree and the symbols, and then we're going to go work through one, and I'll give you some examples. Pedigrees are oftentimes used, pedigree. A pedigree, kind of like what we talked about with Punnett squared, show the possibility of outcomes of offspring. This one is really used more by geneticists and genetic counselors. It basically is a diagram that traces a trait. And again, we're not talking high blood or hair color. We're usually talking things like diabetes or Tay-Sachs disease or whatever that run through a family. So a pedigree is a diagram that traces a trait, usually a disorder, through a family. Now there's some symbols that we need to talk about that you can draw here, and then, like I said, on the back side at the bottom, where we have some space, we'll go through a pedigree and give you some examples of what one would look like. All right, so in a pedigree, circles are representing females, and then guys are square. Sorry, squares are males on a pedigree. Now, as far as the trait and how we represent the trait, so this is the gender representations. Now the trait, I'm just going to use circles, whether they're squares, it doesn't matter. So if it's completely unfilled, like that, they don't have the trait. If it's half shaded in, they carry the allele for the trait. So that one particular trait that might be recessive, that might be missing, or may not be shown here, but that person has the allele for that trait. So they could pass that on to their offspring. And then finally, completely filled in shows the trait. Has it. All right, so this is the shading of the trait. So if I asked you, if I label these number 1, 2, and 3, which ones of these have the trait? 1, 2, or 3? No, have the trait. Just 3. How many of these carry the trait? Carry the allele for the trait? 2 and 3. Okay? There's going to be a question in your homework that's going to make sure that you understand that. Remember, that one question on that Punnett square, what percentage of the offspring carry the allele? When it's carry, it means has it to live. May not necessarily show that trait, but carries the allele to pass on to offspring. So if your heterozygous, big little, you've got the allele. Just because you don't show it, you have the allele that you can pass on to your kid. Okay. Now, turn to your back side of that paper. So if, for example, what a pedigree might look like is something like this. So here's the dad, and here's the mom. The line in between represents a marriage. In other words, these two people are going to produce offspring. Now, the way that you represent an offspring, or offspring on a pedigree, and by the way, this would be parent generation. Remember the first generation up at the top. We'll do that little concept there. The way that you represent offspring from this marriage is a straight line down, and then a line across, and then a line down for every one of the kids that they have. Generally speaking, the oldest starts at the far left. We work left to right. So if I did this, who's the oldest? Boy or girl? Boy. So they have one boy and how many girls? 3 girls. All right, so this would be a what generation? F1. Now, say the baby gets married first, right? And they have a couple of kids, a boy and a girl. I've seen on the SOL, and by the way, that generation would be what generation?
F2.
F2. How many generations are represented on this pedigree? 3. There's 3 levels. Parent, the F1 and the F2 generation are listed. I will also see on the SOL a question like the following. How many kids did the parent generation have? Every year I see is choice A, because it's the red herring, 5. What's wrong with that answer? Did they have 5 kids? No, they had 4. That's an in-law. So be careful when you're rushing and things like that and make stupid mistakes. If you're one that has a tendency to do that, don't. Remember, only the ones that directly come off the parent are the actual children. This is a marriage. Okay. Now, is this possible. Now let's talk about the traits that we've talked so far on in autosome. Is this possible? Yeah. Right. This is possible. Remember, if this is a visual way of looking at big A's and little a's and such, so this would be like a big A, little a, this would be like a big A, big A. So this daughter could have half shaded. She could have gotten half the shaded from her father and the unshaded from her mother. Right? That's possible. Is this possible? No, of course not, because the mom can never give the other shaded in half. Only one half could be shaded in ever. Okay, mom's got to give the other half. It's like the big T, big T. Do they ever give the little T to have a non-taster? No, because mom's always going to give the big T. All right, so that would not be possible. So those are the kinds of things. Or what is possible offspring that possibly happen, whatever. Those are the kinds of questions you would have about a pedigree. So on your test, you're going to practice a little bit for your homework tonight, but on the test, I'm going to give you a pedigree and ask you some questions about it. You might see them pass away. Okay? Any questions about pedigrees or karyotypes? All right. Let's go back to the last two words. Sex-linked traits. Sex-linked genes or sex-linked traits are those traits linked to that 23rd set. Now, this is going to make things a little different. These are traits linked to a sex chromosome. Generally speaking, we only refer to the X chromosome. Why? Because only guys get the Y-linked ones, because only guys get Ys. There are not a whole lot of genes on the Y anyway, just genes for things like, ear hair and Adam's apple and other things that girls don't have. Anyway, there are some disorders that are linked to the Y chromosome, but again, girls don't get them, so we don't really talk about it. Now the thing is that once I start talking about sex-linked chromosomes or sex-linked traits, what? Question? Anyway, the two main sex-linked traits that we talked about, or we will talk about, are colorblindness and hemophilia. There are others, but those are the two most common, I think, that you would recognize and that we talk about. So it's funny because once I mention sex-linked traits, as soon as I mention a blue-eyed man and a brown-eyed woman get married, suddenly it becomes a sex-linked trait just because I talked about a man and a woman. Remember, you'll know, and you'll see an example in a minute, you'll know that it's a sex-linked trait. Sex-linked traits are linked to the X chromosome. Has an X in the name, it's got to be on the X. Okay, that's the only time. Colorblindness, hemophilia are the two main culprits. All right. We'll get to those in a second. Normal ones. Let's take to my least favorite biology word. I usually talk about my favorite biology words. This is my least favorite. My dad was an English teacher. I'm an English teacher's daughter, so I hate double negatives. Non-disjunction. It is disjunction. Disjunction means to separate. So if you're not disjunctioning, you're not separating. Now remember, we just talked about meiosis last class. When do the pairs of alleles come apart? And phase 1 is supposed to come apart because we've got to cut the number of chromosomes by half. It's the number of holes in meiosis. So that our gametes have half the number of chromosomes, so they come together and place half. Except for this. Non-disjunction, when that occurs, those pairs, or a pair, doesn't separate like it's supposed to during N1. When a pair, when a chromosome pair does not separate during N1. And that is going to lead some abnormalities, chromosomal number abnormalities. Now, if you do not have any more room in the back of that page, you're not going to grab a second sheet of medical paper because I'm going to talk to you and diagram some of these abnormalities, chromosomal number abnormalities. Okay, so we're going to go to the non-disjunction problems. Now, my first issue, and I'm just going to draw this is
germ cell is going to become an egg cell. And I'm just going to draw a couple, maybe 3 pairs of chromosomes. As far as autosomal non-disjunction issues, the most common one, there are others, there's lots of others, but I think the one that you'd be most familiar with is when the chromosome pair 21 does not separate. Now, if this is becoming an egg, or sperm, actually, it doesn't really matter. If I'm making my gametes during meiosis, this is round 1. I'm not going to go through all of round 2 and all that stuff. I'm not going to do that. This is round 1. Remember, our pairs are supposed to separate during ana 1. And here they did fine. But for some reason, and it's apparently something going on with the spindle fibers not connecting correctly during meta, both of these chromosome 21s go to the one gamete, and nothing goes to the other. Now, generally speaking, when you're missing a chromosome, a lot of times that baby does not even start dividing. So if this were to be fertilized and become a zygote, it wouldn't. In fact, I would almost guarantee that a woman who has that situation probably wouldn't even know she was pregnant at all, not even realize it, and just keep going. Because nature has this way of understanding that that's not going to work out just right. Something's missing, and that would not be a good thing. On the other hand, on this one, if this one happens to be fertilized, and you have an extra chromosome 21, that's the technical term for that is tri for 3, somi for chromosomes 21. You may recognize it as Down syndrome. No, you have an extra chromosome 21. It's trisomy 21. What that looks like is this. So if you were to have that amniocentesis, I personally was asked if I wanted it, and I said no, because I was older when I had my children. And I said I get what I get. Because the thing is when you see this picture and you see 321 chromosomes, you know that child has Down syndrome. Period. You don't know the extent to the symptoms of that child. You just know that they have it. Anything for something very mild, something very severe. You have no way of knowing. You just know that they have it. And that's a decision you have to make. So that is what a karyotype will show you. Now, is this trisomy 21, this Down syndrome kid, is it a boy or a girl?
It's a boy.
It's a boy because there's an X and a Y. You got a Y, you an I. Easy way to remember Down syndrome is trisomy 21, 3 chromosome 21s. If you were to play blackjack for 21 and you got a king and a queen and an ace, 3 cards, you've got 21, right? You throw your cards down. Down syndrome is trisomy 21. You've got 21 with 3, you throw your cards down. Okay? That's our most common autosomal. There are others if there's an extra 10 or 15 or whatever. I think there's another one where there's an extra 15. I can't remember what it's called. But now we're going to talk about when there's a mess up in sex chromosomes. Okay. Now, next scenario. This is a developing egg. And now this female's sex chromosomes, her 2 X's, stick together for some reason. There are 2 options here. That one went fine, that one went fine, fine, fine. Both of them went to this gamete. Nothing went to that gamete. Now, if that egg on the right, with no sex chromosome, is fertilized by a Y sperm, nothing happens. Doesn't even start dividing. There is no such thing as a Y nothing or a YY baby. There's something on the X chromosome. I mean, everybody's got one. If you look at XX and XY, they both have an X in common, right? Right. So there's something on that X chromosome that's necessary for life. There's no such thing as a Y baby. Nothing ever develops. Now, if that were, however, to have been fertilized by an X sperm, that's a possibility. X nothing can survive. It's called Turner syndrome. Male or female? female. No Y, no guy. Turner syndrome is always female. Turner syndrome, way to remember this one, females are supposed to have 2 X's, the second X got turned away. Turner syndrome. Again, with these chromosomal disorders, with the sex chromosomes, generally speaking, you don't, unlike with Down syndrome or some of the other autosomal traits, you don't generally tend to see any outward symptoms. There's no effect on intelligence or appearance. There can be, but it doesn't necessarily equate to that. Generally speaking, people that have any of these sex chromosome disorders don't generally find out until they try to have children and they can't, because all of these guys are probably going to be sterile because they have the wrong number of chromosomes, some things aren't going to match up. So they try, finally get some genetic testing, and that's when they find out, generally speaking. I did have a female many years ago. She said, yes, I'm Turner's. You wouldn't be able to tell. She was a little bit, if anything, a little of a shorter female, and she wasn't, and of course, remember, she was in high school. She might have developed more later, but, you know, didn't have the curviness of some of us ladies, and of course, some of us have more curve. That's because of our sex, because we eat too much, but anyway, not that kind of curve. But you couldn't really tell that I was shocked. I was like, I didn't know. You can't tell that people outwardly have one of these symptoms. So that's Turner's, and that's the only one where there's nothing in an X chromosome hits. I will fill questions after I'm done recording, just in case they're not appropriate for this particular lecture. Now, this one, where they both stay together, if it is fertilized by the X sperm, that is 3 X's, that is a super female. Again, there's a technical term for that. I don't remember what it is from the top of my head, like trisomy 21. Again, you generally don't see any outward symptoms. This female will not know that she is super female unless she tries to have children and can't. If there's anything with the extra X chromosome, there might be a little tendency to be a bit larger, taller, bigger, you know, maybe a little bit more emotional, a little bit more erratic, possibly because of the extra X and the extra female hormones. But, you know, again, there are females that have hormones and don't have any symptoms during that time of the month and whatever. So, not necessarily a symptom associated with the super female. However, if we do Captain Cranky at the end of the year, which I think we will, there is a super female, and she is a little bit more emotional, but we'll get there later. All right, super female. The other one, if this was fertilized by a Y, I remember this smaller. Fertilized by a Y sperm, it's called XXY. There's an extra X chromosome here. Male or female? Male. You got a Y, you're a guy. And the extra X is called Klinefelter's. Klinefelter syndrome. The way my team teacher helps kids to remember is that extra X that they've got, that's the hallmark of Klinefelter's. If you wrote it in a funny way, it kind of almost looked like a K. X, Y. Remember, that's Klinefelter's. And again, you don't have any generally outward symptoms of Klinefelter's. It's not what you would think. If anything, they tend to maybe be a little bit more boyish looking for the rest of their lives, not as much facial hair or whatever, but for the most part, you can't tell. My team teacher has known several people that were Klinefelter's and didn't find out. One found out as a student because they were doing genetic testing on the whole family for something else, and they happened to find out that he had an extra X. But again, you generally do not tell outwardly. There's no effect on physical appearance or intelligence in most of these guys. Now, last but not least, there is a possibility that the egg is fine, but the sperm, something happens, and yes, DNA replication can, every once in a while, happen during gamete formation. And if that Y chromosome happens to replicate itself and there's an extra Y, there is a possibility of an X egg getting fertilized by a Y sperm. And that, like the super female, is called the super male. And I happen to mention this only because this is one example of science done poorly. We try to show you examples of history where science was done well, with the exception of perhaps Watson and Crick, who pretty much hated everybody. Sorry. These guys, this study was a famous study that was not done right, and you'll see what I mean. Back in the 60s, there was a group of scientists that had a propaganda. They had an agenda. Their idea was that any XYY males should be removed from society because, let's face it, they have an extra Y chromosome. They have extra testosterone. All these guys are going to be more aggressive, bigger. they're going to be all criminals. I mean, heck, the extra Y without that kind of symptoms, it's like the NFL. But their point was these guys should be taken off the streets because they're going to become criminals. Guess where they took their sample from? Jail. Of course, they were already in jail. That was a little biased if you don't. Oh, was that a good sample? No, that was not. I guarantee you, if they sampled guys outside of jail, they would find as many, if not more, XYY males in the outside population. But they only took their sample from in jail. They already were criminals. They were already in jail. And that particular study and all of its propaganda was debunked and thrown out as pure science. So there's an example of a badly done science experiment. Okay, questions about the chromosomal disorders when there's a wrong number. All of these are caused by what happened to cause the wrong number of chromosomes? My least favorite word? Non-disjunction. All right, guys, I want you to turn to page 34. And now we're going to turn to the idea of doing a sex-linked trait cross. Our third twist on our genetics problem. So now we've done the content portion of this lecture. We're going to talk about how we do the genetics problem. Okay. So this is our classwork. So your homework, by the way, to practice the content part of pedigrees and sex-linked traits is going to be page 22. And then we're going to do number 11 for homework as far as the problem is concerned. So 10 is our classwork, for example. All right, now it says, use the following information for the sex-linked cross below. There's your hint. Link to the X chromosomes. I gave you 2 hints. And again, remember, I will not give you a sex-linked trait other than hemophilia or color blindness. Okay? So now it says, normal blood is dominant to hemophiliac blood. Hemophilia is linked to the X chromosome. Remember, sex-linked traits, sex has got the X in it, it's only going to be on the X chromosomes. Because the Y is not the same thing. We'll see that in a second. Do all the steps. Again, needing only one key. Okay, this used to be a worksheet where there's more than one problem based on this. So hemophilia, let's use H's, capital H, lowercase h, look fine for hemophilia. Your homework is about color blindness. Please make sure you use that in your key. And don't be picking C's. As we found out from Carol, Juanita, and Bill, C's are hard to determine big ones from little ones. So go ahead and use big N's and little n's for normal vision and lowercase n for the color blindness on your homework. So, what's going on here? Why does my key look different? Well, this is a special case. When you're talking a sex-linked trait, on the genotype side, it's not enough just to have the letters, the H's or the N's. You also have to have the chromosomes. Kind of like we do with the blood typing in the eyes, you need the X's and the Y's on the genotype side. And then on the phenotype side, it's not enough to say whether they were normal blood or carrier or whatever. You also have to tell me whether male or female. Keys for a sex-linked trait are not worth 1 point, they're worth 2. Because there's 2 things you have to do for each side. Okay? So it looks something like this. If I were to do the visual effect of this, off to the side you might want to draw these. Sex-linked traits are you could use the symbols too if it's a visual thing, you want to do that. For girls are kind of easy because it's kind of same old what we're used to. We've got 2 options for girls, XX, XX, XX. So for normal blood, so you've got X big N, X big N, X big N, X little n, and X little n, X little n. Just like before, big, 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 little, little, little. Look familiar? Jace? And how would we color those females? X big N, which is her title is normal female. And you can abbreviate theme for female. Please do not be using those symbols with the circles and the pluses and the arrows and stuff. I don't remember which one's which. So don't be doing that. And she's completely unshaded in our circle. Okay. X big N, X little n, she's got a special title. She's called a carrier female, and she's going to be the one that's half shaded in. Now, does she have normal blood? You betcha. Both of these ladies have normal blood and normal vision if it was color blindness. They appear outwardly normal, but she's special because she carries the possibility of passing that on to her children. When you're talking something like hemophilia, which, by the way, you know what hemophilia is? Hemophilia, and it used to be a big problem back back in the 1600s and stuff because royals would only marry royals and that allele would stay in the family and it wouldn't spread out. But basically hemophilia is back to DNAs to RNAs to protein, don't forget that, right? There's a problem with the genetics, the chromosome. You got the wrong combination and you make the wrong kind of blood clotting factor, which is a protein. And they don't clot their blood like we do. So they have trouble clotting the blood, making bleed. Normally speaking now, like allergy shots you can get to help boost your antibody production against allergens. They can do this to help them produce blood and clotting factor on their own. Now, I had a student in one of my other classes, her great-grandmother has hemophilia. Now she lives to being a great-grandmother, so obviously these people can live a normal life. However, they got in a car accident and all bets are probably off. Something like that. They've got a major trauma that's probably a different story. But anyway, that's what hemophilia is. So obviously, if a female carries that allele and can pass it on to her offspring, that would be a bad thing. Later on in the year, I hope to show you a movie called Lorenzo's Oil. It's one of those movies that pretty much nobody has seen because it's a serious movie. It's like osmosis jones or something silly. And that is a sex-linked trait. And you see not only the idea of reviewing the genetics and the biochemistry of proteins and stuff like that, and lipids and things, but you actually get to see what would you feel like if you're a mom and you found out that you just passed on this bad X to your boy and he's got a deadly disease that's going to kill him for 2 years. How do you think that feel? How do you think the dad feels about that? So you actually see human aspects about all this stuff that we're covering. There's a point to all this that we're teaching you. It's not just learning how to do research or learning some biology. It's like, yeah, this has real impact on people in their life, believe it or not. So carrier females are special. They do have normal blood, they do have normal vision, but they carry the possibility of passing that on to their offspring. And of course, X little n would be a hemophiliac female, or heme for short, and she would be completely colored in. Now, why are there only 2 guys? Why are there 2 options for guys? Well, there's only 2 options for guys because he could either have the X big N Y or the X little n Y. Why doesn't he have an N on Y? Because that gene ain't there. It's a completely different chromosome. XY is really not homologous. There are different genes on that Y. There are no N genes on the Y. You don't put an N or an H or whatever on the Y. Big sex chromosomes, X, only on the X's. So with guys, they have a greater chance of getting a sex-linked trait. Why? They get one bad X, boom, they got it. With females, both of them have to be recessive. It's a question on your homework. Think about that. With females, they have 2 other options available to them. With guys, they either get it or they don't. 1 out of every 10 males have color blindness in the United States, and 1 out of every 100 females do, because both X's have to be recessive for female. So this would be a normal male. Normal male, and he would be unshaded. And this would be a hemophiliac male, and he would be shaded in. There is no half-shaded square on a sex-linked trait because a guy doesn't get 2 X's. Oh, interesting side note. How many people have heard of a calico cat or have a calico cat? Female. Have to be female. And why? Well, with the calico cat, with females, and this happens to even human females too on some traits, you got 2 X's. How many do we need? How many do guys have? 1. You really only need 1 X. So girls, ladies, we have an extra X basically. Okay? So what can happen in certain situations, in certain cells and whatever, 1 of the X's can be inactivated, not function. And that's fine because the other one is fine. In calico cats, on the sex chromosomes, that is the gene for their hair color. So there's a black X and there's a red X or brown X. So when the black X is inactivated, those are the spots that are brown. When the brown one's inactivated and the black one's active, that's the spots that they have black. So a calico female, calico cat has to be female because you have to have the 2 X's. If you ever find a calico cat that's a male, it's worth a lot of money, except the fact that it probably won't reproduce, so don't think about selling that thing because it's not right now. Okay? So here we have a sex-linked trait key completely done. Have to have not only the letters, but the X's and the Y's on the genotype side. You have to have the trait and whether it's male or female. And don't be putting stuff like bracket female, bracket male. Go ahead and write it out. I know things were done, but you need to get the practice of this. Now here's a problem. How do we put this all together? So you will model doing that, except again, I used ends, didn't I? You could use the ends or just whatever. I don't care. As long as you use letters that are different looking. All right, so now a problem would be something like this. Cross a carrier female with a hemophiliac male. All right, so carrier female is X big N, X little n, and a hemophiliac male would be X, I'm back to my H's, sorry, X little n, Y. Does everybody see that? Okay, all right, so now I'm going to go ahead and put them down in my problem. X big N, X little n, X little n, Y. Now, bad news is the keys are pretty crappy, and the ratio is going to be pretty crappy because we write everything out. Good news is it's always going to be a 2 by 2 Punnett squared because we only have an X and an X. So one gamete could be the X big N X or the X little n X. For the dad, X little n or the Y. Guys, you got your X's from whom? Excuse me? Your mom. You got your Y from your dad. You got your X from your mom. So if you have an X-linked trait like bell-patterned baldness, blame her because she gave it to you. Something silly like that, that's fine, but something like ALD, not so fun. All right, put your parents in the gametes, whatever's in the circle, put them there. I don't care what parent you put where. I usually put the female, the mom on the top, and dad to the side. Doesn't matter. So X big N, X little n for mom. Check, check. Dad is X little n. Check, and Y. Check. You could put dad up here, X little n, Y up here, and X big N, X little n for mom. I don't care. It's going to turn out the same way. Now we got to put them together. So this one, now when you put them together, just like before with the superscript of the letters, put the capital letter first. So this would be X big N, X little n, and X little n, X little n. And the boys, X big N, Y, X little n, Y. And yeah, the genotype and phenotype ratios stink, but I want to point out something. All right, so genotype ratios, I've got 1, X big N, X little n, 1, X little n, X little n, 1, X big N, Y, 1, X little n, Y. Now, if this had been a normal female, big, and this one was a carrier female, big, little, every time for my phenotype ratios, I got 2 normal females. One's a normal female, one's a carrier female. They have separate titles. Now here, it's an issue. I've got 1 carrier female with normal blood, 1 hemophiliac female, heme, 1 normal male, and 1 hemophiliac male. Couple questions. Of the children, so of the children, remember these 4 boxes are all 4 children possible. How many could be hemophiliacs? 2, one of the girls and the boy, one of the boys. Of the boys, remember it's the boys, how many could have normal blood? 1. Just 1. Of the children, how many could have normal blood? 2. Remember, the carrier female also has normal blood. Be careful. Titles versus what they have. In fact, a lot of times what I will do is to remind myself that yes, she's a carrier, but she's got normal blood, and he's got normal blood, or normal vision, normal vision, normal vision. Because I might ask a question, how many are normal? That's the title. Normal vision or normal blood, that's the outcome. So be careful when I ask that. Your homework is a little tricky. I'm not going to give you any help. This one, I give you clues. It says colorblindness. So again, use the ends. There's another X-linked trait. That's our other sex-linked trait. 2 parents with normal vision have 4 children, 2 girls and 2 boys. Both girls that they have normal vision. 1 boy is normal, 1 boy is colorblind. What do the genotypes of the parents have to be to have these kids? So you're going to have to work backwards. Figure out what the kids are and then figure out what the parents are. It's a little bit of a puzzle, but you can do it. First thing you do, as usual, is do your key. Then, I would do your Punnett squares, put your kids in, fill that out. Given the clues that I've given you, both parents have normal vision, 2 girls, 1 normal, both of them have normal. boys, 1 normal, 1 colorblind. And then go back and fill out the rest of the sum. Okay? Any questions? Next class, we're going to review stuff, and I'm going to do some of those tests with you. We're going to test you for color blindness and for tasting, just for fun, to see what that is. Sort of, kind of. And then we will field any last minute questions. Hopefully we'll be doing that lab. If not, I'll figure out something else to do in lieu of that. And then on Friday, I'll assign the trade and grade next class, which is the last 2 pages. And then we'll review for the test on Friday, test is on Tuesday. Now on the test, since DNA to RNA to protein is kind of linked to this, because it's how we use that, there's going to be a couple of questions, about 5 or so, that have to do with DNA and all that stuff to review to lead into this part as well. So we'll talk about that more on Friday. Okay? So you guys have about half an hour. Go ahead and work on your 2 homeworks. If you have any questions about